Hello, my name is Andre Lagron, and I will be presenting on the relative toxicities and mechanism behind the toxicity of our favorite substances, the nerve agents. So they are listed here in order of toxicity. Here we have the G series, and here we have the V series agent that we really care about. So here we have tabun, sarin, and then soman. That is the order in which they were discovered and the order of their toxicity. VX and Soman, this is a contentious relationship as far as which one is actually more toxic. VX is more immediately toxic than Soman. The acute symptoms happen much quicker. However, Soman undergoes a separate process. The secondary process, the aging process of the nerve agent, which makes the effects permanent. And Soman undergoes that process faster than any other nerve agent. So Saman is far more dangerous if you are prepared and you have access to atropine and 2-PAM-CL antidotes. However, VX is far more deadly for unprepared civilians or troops. So that's why, in my opinion, and the general consensus, is that VX is the more toxic of the two. Now, the Novichok agents, they sit somewhere in this range. So the reason behind that is that it is a class of agents. It is not one specific agent, and so they have a variety of toxicities, and we don't really know that much about them. We don't know how many there are. We don't know what they are, what they do, even the structures of really any of them. So for this reason, I won't be focusing on the Novichok agents, and by the way, those are things like A230 and A232. So we won't be bothering about the Novichok agents. We're going to be focusing on our favorite four over here. So let's start, as all things do, at the beginning, which in this case would be tabun and the mechanism of cholinesterase inhibition. So let's begin. Here we have the structure of acetylcholine, which is the neuroreceptor that nerve agents emulate. This is my lovely, incredibly detailed drawing of cholinesterase. This is the serine active site. That's the specific amino acid at the active site that undergoes the reaction. So acetylcholine comes in, and this is the anionic site. So what happens is acetylcholine, this nitrogen will be attracted to this anionic site, and it'll electrostatically be drawn to it and kind of hang out there. Meanwhile, this lovely reaction will occur and basically lead to the breakdown as this ester attacks the alcohol group. It will lead to the breakdown of acetylcholine into acetic acid and choline, which I can draw here. There we have acetic acid, and choline is this structure here. So these are the two parts that acetylcholine breaks down into via an ester hydrolysis reaction at the serine active site, with, very importantly, this bulky nitrogen group being drawn towards this anionic site. So what do nerve agents do? Well, they mimic the structure of acetylcholine. And effectively what happens is this structure here is, let's see, starting here. This structure is drawn to the anionic site. The phosphorus will form a bond with the oxygen here, and it will yield the following product. So this is the product. We wind up with a covalent bond here, which prevents any further acetylcholine from interacting with the alcohol group, right? a water molecule will come in here to perform this hydrolysis reaction. And again, note that there is no longer a hydrogen on this oxygen, so we are no longer able to actually perform the acetylcholine hydrolysis. All right, so why the heck would we care about acetylcholine hydrolysis? Let me show you. So here we have an example of a nerve synaptic cleft. So here we have the neuron that is sending the signal, and this is the neuron that's receiving it. So our signal is going in this direction. When the signal reaches this part of the first neuron, it will release acetylcholine through these channels into the intercellular space. And what acetylcholine will do is it will repeatedly bind to receptors. So it'll come in and out over and over and over again, and each time it does, it sends a flood of neurotransmitters within the cell, or signal molecules within the cell to send the cell signal along. Now, that's not good if it's just sitting there, because it'll repeatedly activate the neural pathway. So that's where acetylcholinesterase comes in. What cholinesterase will do is it will pick up this acetylcholine, get rid of it, break it down, and then release the byproducts, leaving the cholinesterase available to pick up the next one and the next and the next and the next. Now what happens when nerve agents come in, it will be this goldfish-looking thing, is they form a covalent bond at the active site, which prevents any acetylcholine from actually being broken down. Because of this, they continue to cycle, sending signal after signal after signal after signal into the cell. Now, when they first bind, it's possible to boot them out with a different substrate. However, once the aging process occurs, it is no longer possible to separate them. And this acetylcholine is dead. It is dead and gone and not useful for anything. It's not technically alive. It's just a protein. It's just an enzyme. But for the purpose of anthropomorphication, this acetylcholine is gonzo. So when the acetylcholinesterase, when it's gone, then we continue to excessively signal within the cell and between cells, and this causes all kinds of horrific problems. So that's why we care about the inhibition of acetylcholinesterase. Now let's get on to sarin. What about sarin? What makes sarin different? So here we see the structure of sarin. Now sarin is structured a little bit differently from tabun. You can notice an important difference here would be the replacing of this nitrogen group and with a fluorine group, and this nitrile group with simply a methyl group. Sarin will more swiftly bond to acetylcholinesterase, and it will age slightly quicker than tabun does, thus making it more toxic. What about soman? Now, soman has a very different structure, although not necessarily as chemically different as one might think. So basically the difference between sarin and soman is the bulkiness of this ether group. And basically what that bulkiness does is, via the mysterious and spooky aging process, which I was not able to find a lovely reaction diagram for. If someone does, please remake this video. I would love to know more about it. Simply put, soman binds a little bit slower than sarin does because of how bulky this group is. However, upon binding, it ages extremely quickly, on the order of 1 to 2 minutes. So that makes soman far more toxic. Now, what about VX? What does VX bring to the table? Now, what, just by looking at this, can we tell would make it so that VX is far more toxic? If we look at the structure of acetylcholine, we can see that the structure of VX is actually extremely similar. Right? We have this sulfur replacing the oxygen, and this nitrogen has bulky isopropyl groups instead of 3-methyl groups. But in terms of overall structure, it's extremely similar to acetylcholine. And this allows it to bond incredibly quickly. VX is unbelievably fast at inhibiting acetylcholine esterase. However, it doesn't age it as quickly as SOMAN does. So quick application of PAM and atropine will more effectively take care of VX poisoning than SOMAN poisoning. However, because of VX's extreme toxicity and how quickly you'll have the immediate acute effects, which, include things like suffocation and dying, it is far more dangerous to civilians than SOMAN is. So basically the way to remember this, SOMAN is more toxic than VX, but VX is far faster. More toxic, far faster. The way I remember this is if you have the name SOMAN, and you get rid of that A and that N, you add a T at the beginning. It's the word most backwards. It's the most toxic. I don't know, it's 3 of the letters of the word most in it. And VX sounds like the model of a car that someone is desperately trying to convince you is extremely fast. So, SOMAN, VX faster at killing people. SOMAN is better at it, if that makes sense. So, or SOMAN is at least more permanent. And that's what I have for you. This was something that I was wondering about. I really would love to have had broken down more in class, so I made a video about it. Enjoy! Good luck!